Hello, welcome to the fourth part of the gene expression unit. In this lecture, we will evaluate how we identify proteins based on tandem mass spectral collections. So in the prior video, we talked about how tandem mass spectrometry creates these tandem mass spectra that represent the fragments of a broken peptide. So we have an intact measurement of the peptide, the intact mass, and we have the fragments that were produced from it. That's a tandem mass spectrum. So to produce these data, we went through quite a lot of steps on the bench. We started with a mixture of proteins, we digested them, we then separated those peptides in time by chromatography and produced tandem mass spectra representing the fragments of individual peptides. That's the trace down the left side of the screen. At the next stage, though, we need to do a lot of these steps in reverse, in silico, in the computer. So for the first step, we need to be able to rematch a tandem mass spectrum with the peptide sequence that gave rise to it. That's the province of the database search algorithm. That's the heart of today's lecture. The next stage is that we need to figure out which of these matched spectra are confident matches and separate those from those that we feel much less confident about. That's the next step that we see here. In this case, we'll talk about primarily the target decoy method for applying a false discovery rate to the identified peptides. And finally, we need to reintegrate the data at the protein level to say which proteins are the best hypotheses for explaining the peptides we've identified. So the database search algorithm is kind of the key piece of property in the intellectual property of bioinformatics for proteomics. We start by looking up the answer in the back of the book. In other words, we're going to start from what list of proteins are possible for the species that we digested up to produce this set of tandem mass spectra. So if we started with a clinical sample, we're going to start with a human proteome, the full set of all human proteins that we know to exist. For about 20,000 genes in the human genome, we assume at present that there are something like 110,000 proteins that can be derived. Remember, multiple transcripts may come from an individual gene, so multiple proteins are possible for a given gene sequence. So the FASTA sequence database is going to provide us the list of all proteins that are possible for this species. From that, we'll perform an in silico digestion, so emulate the behavior of a digestive enzyme like trypsin to chop up that protein sequence at every arginine and lysine residue, because that's where trypsin cuts. So that gives us the list of all peptides that are possible from this species. Next, we're going to do something we call the PTM expansion. The user may have specified that particular post-translational modifications are possible. One of the most common ones that we look for example, is the oxidation of methionine. Methionine is just very prone to getting oxidized when you have a sample digested up. So we are going to consider the possibility for each amino acid that's modifiable that it is modified or it isn't modified. That's going to create a little fork in the set of peptides that we have to consider. So that decorates the peptides from this organism, allowing us to consider every possible combination of modified and unmodified for the specified post-translational modifications, not for all possible ones. That would be far too big. Now we have to figure out which peptides can be compared to this particular spectrum. This is something called the peptide mass filter, and we're going to talk about that in the next slide. So once we know which set of peptides can be compared to a particular tandem mass spectrum, we need to predict for each sequence that's possible, what should the spectrum have looked like if this were the peptide sequence that broke to produce these fragment ions. So this is a prediction of what the spectrum should look like for a given peptide sequence, and then that projection of what the spectrum should look like can be compared to the spectrum that we actually observed. And that's done in a match scorer. This was all introduced back in 1994 and 1995 by two papers from the Yates lab. That's where I did my PhD, so this has been something that I've cared about for a long time. So what is a peptide mass filter? Remember, we were saying that we have the full set of all peptides that could result from this particular species. How do we decide which ones get compared to this particular tandem mass spectrum, this particular collection of fragments? Remember, we didn't measure the peptides just by the fragments they produce. we also measured the intact masses of the peptide ions. So this is great, because now we know that if we know the intact mass, how massive this peptide was before it broke apart, we need only compare those sequences that are very, very close to that observed mass of the peptide. So that's brilliant. Now we have an observed mass for this peptide, and we know that we must be within some distance from that observed mass with a computed sequence. Now, as you can see at the bottom, different instruments that we use for tandem mass spectrometry differ in the mass accuracy of their observations. So if we've run this experiment on a Fourier transform mass spectrometer, probably the Orbitrap is the most commonly used type of that class, then we probably know the mass of that intact peptide within 10 parts per million. That's an astonishingly accurate measurement. You know well better than within a single neutron. You know within, say, 0.01 m/z to either side. That's a mass to charge to either side. That's an incredibly accurate measurement. It may be that we used a less accurate mass analyzer, like a time-of-flight instrument. Those we are also quite accurate, but generally they're above 10 parts per million, maybe as bad as 100 parts per million in some cases. Or it could be that we had a very inexpensive mass spectrometer called a quadrupole ion trap. These measure mass less accurately because they can't even differentiate all the isotopes that are produced for a given peptide. So in a case like that, we may not know the mass very accurately at all. We may know it within, say, 1.25 mass to charge units, Daltons per charge. So depending on how expensive your mass spec is, you may have a lot of sequences to compare to each spectrum or just a very small number. Okay, so we've now decided on a set of peptides via the peptide mass filter that will be compared to the spectrum. But how do we do that comparison? In the Sequest algorithm, I'm giving a technical explanation in the slide, but I hope you'll just follow along with the simple one, basically. We have a projection of what the spectrum should look like if it were the sequence, and we have the observed spectrum itself. What we're going to do is rub one past the other at different offsets and ask how well do they align at each of these offsets. Now at the bottom right of the slide, you can see that there's a spike right at 0. So when there's no offset in their mass values, that means that an ion we projected at 500 matches to an ion that was observed at 500. So what we see is that when the correct sequence is compared to the spectrum, we have a much better alignment at 0 offset than we do when we have just a very slight offset in either direction. Okay, so that Sequest cross-correlation algorithm is kind of the great granddad of all of the search engines that are out there. There have now been more than 30 database search algorithms published in the peer-reviewed literature, but Sequest is still used, and this scoring approach is still used. Most people today might use the Sequest algorithm from Thermo, licensed from Thermo, or they might use, for example, Jimmy Eng's later software called Comet, because they score exactly the same way. So this is an approach that's been quite durable over the last several decades, a couple decades. Now, there's also a statistical way that we can think about this. Imagine that our spectrum is instead a jar of marbles. And maybe there were 1,000 different places that might have held a peak in that spectrum, but only 100 of them were occupied by a peak. So that jar of marbles is going to have 900 white marbles to reflect locations in the spectrum where there might have been a peak but wasn't, and black marbles that indicate locations where there might have been a peak and there was. Okay, so 100 peaks in a spectrum that was about 1,000 peaks in size. Now, I don't care about all the jar, the full jar of marbles. I only care about these few locations along the mass to charge axis that should have had a fragment if this sequence were real. Okay, so for that case, we pull out the marbles that correspond to just those locations in the spectrum, and we ask, how many white and black marbles do we have in our hand compared to those that are in the jar? Now there's a little bit of math down here at the bottom that explains that. We had 100 black marbles in the jar, 900 white marbles in the jar. That's a total of 1,000 marbles. In my hand, I had 15 black marbles and 5 white marbles. Now we can ask, is that likely to have happened by random chance alone? The hypergeometric distribution lets us compute the probability of that particular number of black versus white marbles in our sample, in our hands, those that associate with the sequence. And in this case, you see that it is an astronomically small probability. So this is much more likely to be a correct hit, a correct explanation for why the spectrum looks this way than a random sequence drawn from a database. Okay, so that's a statistical approach for doing the same thing. So we have now produced what we call a collection of PSMs, peptide spectrum matches. We started with 10,000 spectra or 1,000,000 spectra, however many it was, and for each of those spectra, we've got some individual sequence that scored best out of all of the database. But we want to know which ones we actually trust. There have been two different approaches produced, two general broad approaches that have been used in this case. One is a distribution modeling approach that attempts to say, what is the probability that this individual PSM, this individual peptide spectrum matches right? This is something that was produced most prominently in the peptide profit model that you can read about. I didn't include the citation, but you can, it's cited in the paper mentioned here. There's also another approach called the target decoy approach that sorts all of the spectra from the best score to the worst, and for every place where you might place a threshold against that list, you ask, what is the false discovery rate for the general pool of identifications that pass under these circumstances? So that's a much broader technique that is only intended to estimate the error rate for the whole collection of past PSMs. Now, the final piece I really want to talk about is that we need to be very careful in claiming proteins are identified, because what we measure in tandem mass spectrometry is a peptide. Each tandem mass spectrum in this conventional approach represents an individual peptide, but we do not directly observe proteins in shotgun proteomics. So if we're going to try to infer from this peptide list which proteins we've got, we need to take into account that there are several reasons why an individual peptide sequence might appear in multiple proteins throughout the database. So as you can see in this graph, we have a set of proteins at the left and a set of peptides over at the right, and we've drawn an edge for every protein that can explain each peptide. What we try to do is claim a minimal set of these proteins in order to account for all of the peptide evidence that we've got. Back in the bad old days of the 1990s, we might have been content to say, well, all 7 of these protein groups have evidence in our peptide list. But now we have to be much more careful. We try to use a principle called parsimony, meaning we don't go further than the evidence must take us. So we want to explain every peptide that we've identified successfully. That's true. But we don't need to explain each peptide more than once. So once you've explained a peptide, you no longer need to re-explain it with more and more peptides. So in this case, the 7 protein groups shown in blue on this slide, we see that only 2 need to be instantiated, need to be claimed, in order to explain all of the peptides at the right. So someone failing to use parsimony would overclaim by 5 proteins the number that are required to explain the evidence. So an experiment for proteomics will typically use lots and lots of reverse phase liquid chromatography tandem mass spectrometry experiments. They may produce 1,000,000 or more spectra. So if that's the case, we're going to spend a fair amount of computational time matching the tandem mass spectra to particular peptide sequences. You can't just publish all your IDs. You must have some way to control the false discovery rate so that you know that the peptides you're claiming have some legitimacy. Proteins are inferred from proteomic evidence. They are not directly identified in the shotgun method. Somebody who wants to identify proteins directly in their intact form needs to be working in a field called top-down proteomics, which is actually quite a hot field to work in. So right now, with a given experiment, with a given sample, you can identify more than 100,000 peptides. Typically, that would amount to something like 10,000 proteins from these samples. So the maturity of this field has really come a long way in the 20 years I've been watching this field. So thank you very much for taking part in the gene expression module for H3A Bionet.